dosage form. So as you all know that curcumin basically is a natural yellow pigment in turmeric, and it is isolated from the rhizomes of the plant uh, Curcuma longa, and it contains about 3 to 4% of curcumin, which is the main thing. And the major breakthrough in realizing the medicinal values of turmeric came with the isolation of the phenolics called as the curcuminoids, of which curcumin is the major component. So curcumin has been traditionally used as an antimicrobial agent, as we all know. Like even as of today, if we just get a scratch or if we get there is any bleeding, the first thing at home what we wish to do is we will quickly put curcumin, that is the turmeric that is at home. Okay, so as we all know that microorganisms such as bacteria, they cause many types of skin related disease such as skin rash, acne, eczema, psoriasis and dermatitis, and these are really tough to treat. And Staphylococcus aureus is the main pathogen that cause such skin infections. Therefore, topical dosage forms containing antibacterial properties are used to treat and prevent such infections. Now, several studies have reported the broad spectrum antimicrobial activity for curcumin, including antibacterial, antiviral, antifungal, and antimalarial activities. And gentamicin, you know, is a broad spectrum aminoglycoside type antibiotic which is isolated from the Monospora purpurea. Now, gentamicin kills bacteria by damaging the plasma membrane and binding to the 16S ribosomal RNA, leading to the inhibition of the microbial protein synthesis. And it is effective against wide spectrum of gram-positive and gram-negative bacteria. So the synergistic antibacterial activity of curcumin with antibiotic gentamicin against Staphylococcus aureus have been reported, and it has been concluded that curcumin enhances the antibacterial effect of gentamicin. I hope I'm audible to all of you.
Yes,
okay, thank you. Yes. Okay, so the need for the study, like I told you, is that we had scientific investigation of curcumin, which was performed against 8 different antibiotics. And amongst these 8 antibiotics, gentamicin, ciprofloxacin, and amikacin, they showed synergistic antibacterial activity, which enhanced the activity of curcumin when combined with these 3 antibiotics. And therefore, we chose to take gentamicin and we prepared a dosage form, a topical dosage form. We prepared a cream. Now, there are numerous combinations of herbal with herbal drugs and synthetic with synthetic drugs. But based on the literature study, the combination of curcumin with a synthetic antibiotic was not available in the market. And therefore, it motivated us to prepare a formulation. We optimized the formulation and we also evaluated, and we also came about with a new analytical method for the combination, for the dosage form that we did. So the objective was, like I said, mainly to design the topical formulation for curcumin with gentamicin. We optimized and evaluated it, and we also developed and validated a new analytical method for the estimation of newly prepared formulation of curcumin and gentamicin. I hope all of you are finding this a little interesting and a little different from all the topics that you've had. Am I? Yes or no?
Yes,
yes, ma'am. Yes, ma'am.
Okay, fine. Thanks. Okay. So it's better that we have a little interaction, because somehow I am not able to see you nor myself. So instead of it becoming a very one-sided, I'm just going to have you all in the interaction so that we can make it a little better.
Sure.
Okay. Fine. So now we formulated a topical cream. Now this it was a basic oil in water emulsion based cream using various parameters and excipients. The drug dose remained the same. This is what we did. And like you all know, the cream is like, when you're going to prepare, we are going to have the aqueous phase and the non-aqueous phase, and we are going to mix it together, and the cream was going to be prepared. So what we had is the curcumin, which had the oil soluble components, because curcumin is oily in nature. So it had, with the oil soluble components, like we had stearic acid, cetyl alcohol, coconut oil were taken in one china dish, and in the other we had the water soluble, that is the aqueous, that is we had gentamicin and ascorbic acid, triethylenamine, propylene glycol, methyl paraben and propyl paraben, that is the preservatives that we use. These were weighed, And then further, both the china dishes were then heated in a water bath up to 75 degrees. After heating both, the containers were then taken off the water bath and the water phase was slowly added to the oil phase with continuous trituration until the cooling of the emulsion and the cream was prepared. So we had almost around 7 preparations. That is, so we kept the drugs like you can see. Oh, I'm sorry. Okay, so you will see that curcumin 1 gram and gentamicin 0.1 gram were kept standard. We did not change the concentrations for the same, but we changed the concentrations for the aqueous, that is the stearic acid, cetyl alcohol, and coconut oil. Similarly, for ascorbic acid, triethylenamine, propylene glycol, methylparaben, and propylparaben, and sufficient water was taken, of course. Okay, so these were the thing. Just to tell you now that finally FY was the formulation that was taken, F5, because it had good penetration, because penetration is the most important when you're talking about topical preparations, because better the penetration, better is going to be the contribution of the drug to the skin. So F5 was taken up. Then in this, we again did the pre-formulation studies as usual, as we need to do for all. Then the identification of the drug by spectroscopy was done and DSC analysis. The melting point had to be done, so that was done. Solubility analysis, compatibility studies, determination of the lambda max, and calibration of curcumin with the gentamicin. So these were the pre-formulation studies that we did, which we generally will do it for any other kind of a dosage form that we are going to be preparing. So if you see the IR spectroscopy, I may not really go into the details, but just to tell you that it was performed and the peaks were formed. So the curcumin and gentamicin we took. Then we also took the melting point of curcumin and gentamicin again by the DSC analysis, so that we made sure that the purity of the drug was tested for. Then we did the melting point using the thistles and for curcumin and for gentamicin, and 183 degrees and 240 degrees respectively was taken, and it was matching the theoretical and the literature value. Then we did the solubility analysis. In solubility analysis, curcumin is, since it is oil based, it is insoluble in distilled water. However, it can be made soluble in water by using a alkaline solution. So if you add a phosphate buffer, it can be made soluble. Then gentamicin also, which is insoluble in benzene but freely soluble in water. And the compatibility studies was done by the IR spectrophotometry. So we saw that curcumin and gentamicin, so we took individually the peaks for curcumin and gentamicin. Then we added curcumin and gentamicin together. Then we also took curcumin, gentamicin, and the excipients that we were supposed to be using. And we saw that there was no incompatibility with either. That is, either with the gentamicin and curcumin by themselves, or with curcumin, gentamicin, and the different excipients that we used for both aqueous and the non-aqueous. So we did the compatibility thing. Then we also took the lambda max. So lambda max was taken, and the absorbance of the pure for the pure drug was found. The absorbance spectrum was taken and it was noted. Then, so this is the calibration curve. So you see that the R factor was close to 0.99 and 0.996, which is good, and it is within the limits. Then we came to the evaluation of the cream containing the curcumin and the gentamicin. So once we prepared the cream, then we had to evaluate the cream, because that is the next step of what we were planning to do. So whenever you're going to be talking about the dosage forms, so especially for the topical, then you need to see that the physical appearance. So for any dosage form for that matter, even if you take for tablets, for capsules, and for any topical dosage forms, you need to have the physical appearance. So physical appearance here, that it was yellow orange in color, it had a smooth texture, and it did not have any phase separation. So which shows that it had a good homogeneity, that is the aqueous and the non-aqueous were mixed well. Then we had the pH, that is for the pH of the mixture we took using a digital pH meter. So we took 0.5 grams of the cream, we dissolved it in distilled water, and the pH was measured, and it was found to be 6.40 to 6.90, which is ideal enough to be applied on the skin. Then we did the viscosity with using the Brookfield's viscometer, and the viscosity was between 1442 to 2970 CPS, which was good enough because it helped the extrudability. So extrudability is the amount of the ointment that you cream that we were able to take out of the foil when we added. So viscosity was good enough. Then the drug content percent for the cream, which was supposed to be, which was found to be 97.8% for curcumin and almost 98% for gentamicin, which was again good enough. Then the spreadability of the cream was determined. So for the spreadability, like we took 2 glass slides. So 0.5 grams of the cream was placed on one slide in the center. Another slide was placed over. Weight was placed on the slide, and it was kept over a period of time. When we picked it up, the diameter was taken. So it showed a spreadability of a good 3.5 to 4.6 centimeters, which was good enough. Then the extrudability of the cream was determined, and it was easily extrudable for all the formulations. So for the and then once we did the extrudability, that is, it was filled in a clean lacquered collapsible 5 grams tube with a nasal tip of 5 millimeter opening, and pressure was applied with the fingers, and the cream came out. So for all the formulations we had to try. It was good for all the formulations. However, for the formulation that we picked up, it was equally good. Then the consistency of the cream was found to be smooth, no lumps and no grittiness. So this is the evaluation of the cream that we did. Also, we did the irritation test was done, the in ex vivo, and there was no irritation found. Then, so this is in the table form that we did just now. Okay. Then we also did the in vitro drug release. Now this in vitro drug release. Okay, sorry. Okay, for one in vitro drug release, what we did is we took we used the France diffusion cell. So we took 1 gram of the formulation, and we then placed it in the diffusion cell with a semi-permeable dialysis membrane with a molecular weight cutoff point of 1000. Which was previously immersed in a receptor phase for overnight, and it was placed over the mouth of the receptor post. Then the cell body was filled to the overflowing with a degassed phosphate buffer of pH 6.8 as the receptor phase, and was maintained at a constant temperature of 37 degrees, that is the body temperature, and by means of a water jacket in each cell. Then the donor part containing the sample was then placed over the semi-permeable membrane, and the diffusion was allowed to take place. The receptor phase was stirred thoroughly by constantly spinning a bar magnet at 100 RPM, and at the intervals of 30 minutes, 1, 2, 3, 4, 5, 6, so up till 12 hours, every hour was chosen and as a sampling time, and the samples were analyzed for curcumin and gentamicin at 421 for curcumin and 207 nanometers for gentamicin. A 1 ml aliquot was removed, and it was replaced with an equal volume of the receptor phase. So this way we made sure that the drug content was good, that is the in vitro drug release was optimized this way. Then we did the antibacterial activity. So for the antibacterial activity, we prepared a nutrient agar medium. Then I'm sorry that I put this picture was little wrongly taken, but I was not able to change it in the morning. However, the antimicrobial activity shown was really very good. It was done by the disc diffusion method. So 4.2 grams of nutrient agar was dissolved in 150 ml of distilled water, which was then heated so that the content dissolves, and then it was autoclaved for 15 minutes at 121 degrees centigrade. Or after autoclaving, it was then allowed to cool, but it was not allowed to solidify, which is, that is how it has to be done to form the cup. Later, this content was poured into the Petri dishes, keeping it on a sterile surface, then until it solidifies. Then 150 ml of the nutrient broth media was prepared by using 4.2 gram nutrient, and then after sterilization, it was transferred to the pre-sterilized test tubes. The culture from the stock was then transferred into the prepared test tubes using the preheated inoculating loop, and this prepared the subculture for further and kept for incubation for 24 hours at 37 degrees. So we see that the disc fusion method was adopted to perform the antimicrobial testing, and the prepared subcultures were swabbed into the Petri dishes, and it was allowed to solidify. So here you will see marked, I don't know how clear it is, but then we had G standing for the gentamicin standard, then we had C for the curcumin standard, M is for the mixture of both the standards, sorry, and the F is for the formulation, and B is for the blank which stood. Then you will see that the zone of inhibition for the cream containing the curcumin and the gentamicin against Staphylococcus aureus was, for curcumin it was 7, for gentamicin 10, curcumin plus gentamicin mixture was 9, and the formulation was 10, which is really very good. That is, if you compare, it had the maximum zone of inhibition. So then once we did this, then we optimized the formulation. So optimization of the formulation, that is the antibacterial cream containing curcumin and gentamicin, was prepared using the suitable excipients. And the cream was evaluated for several evaluation parameters, like I said, the physical appearance, pH, viscosity, spreadability, which we have already discussed. Then once we did that, then we went for the analytical method. Now, whenever you are going to talk about this, that is the drug discovery, development, and manufacture of any pharmaceutical drugs are most preferably dependent on the analytical method development and validation. So these analytical methods they help us to identify the challenges in the development and the validation of the pharmaceutical products. Therefore, method development and validation help in the identification of impurities and also help in the development of methods for new versions of drug qualities. So whenever a new dosage form comes, or whenever a new molecule has come into or is going to be taken up for the preparation of a dosage form, we first need to have an analytical method to validate the either the molecule or the dosage form, whatever is going to be in question. So most of the time it is going to be, you're going to validate the pure forms, then you're going to validate it for the dosage form. So for the development of the analytical method form, what we did was the simultaneous estimation was done for curcumin and for gentamicin, and we took many like we had to keep trying, but finally we could do this. And then we took the analytes, that is curcumin and gentamicin. We took water as a solvent, and we got the exact 207 for gentamicin and 421 for curcumin. So which was okay. So here you will see, that is we got the UV spectrum for gentamicin, curcumin, and then we took it for the mixture, and the linearity overlays you can see, which took place at 2,207 and 421. So this validation was done for specificity, and that is a selectivity of the molecule, specificity of the formulation, specificity of the
Ma'am, your voice is not coming. Hello. Hello. Ma'am, your voice is not audible. Hello, madam.
Yeah.
Okay, madam. Now it is audible.
Yeah, thank you. Okay, I'm sorry. The power went off and
Okay,
my
madam. No
internet
problem.
connection was lost. So,
Okay, madam. No problem.
whenever we are going to be talking about the analytical method development, then we need to talk about specificity and selectivity, the linearity, precision, robustness, and the stability studies. So these are the factors that we consider whenever we go in for an analytical method development. So for validation, you generally will go, that is a standard calibration curve was plotted and concentration versus absorbance were taken, and the absorbance were noted. So the LOD and the LOQ were taken, and it was well within the range that we needed. Then we had the LOD and the LOD values of gentamicin and curcumin, which was found to be 0.39 micrograms per ml and 0.38 microgram per ml for curcumin, and the LOQ values for gentamicin and curcumin was found to be 1.18 microgram per liter and per ml and 1.16 micro, and we saw that the percentage RSD was less than 2%, which is the acceptable limit. So, in conclusion, the recent study was attempted to formulate an antibacterial cream of curcumin and gentamicin, which acts against Staphylococcus aureus. And several studies have been reported that curcumin, in combination with this antibiotic, works synergistically against Staphylococcus aureus. And there are no formulations available in the market, which is the most important aspect that we considered when we took up this study. And also the method development of curcumin and gentamicin in the bulk powder has been performed. Also this method has been published. So the paper has also been published for the method development. The FTIR and the DSC are the preferred analytical methods for compatibility studies, and the optimized formulation had the highest release rate compared to the other formulation, and also it had the best permeation. And the UV method developed for the simultaneous estimation of curcumin and gentamicin in the bulk powder. And this research concluded that newly developed spectrophotometric technique was found to be simple, specific, selective, linear, precise, robust, rugged, and reproducible. We also did the precision was taken for intraday, that is, we took the precision for morning, afternoon, and evening. Interday, that is every day for 3 days. The ruggedness was done by using the analyst, and also by changing the instrument, and the stability was checked. And the RSD was less than 2%, which is the acceptable one. So this is the conclusion, and go to the screen card, and let me just see the participant. Yeah. Okay. So this was my presentation for today. If you have any queries.
Ma'am, good morning. Ma'am, good morning.
Yes, good morning.
I'm calling from Chennai.
Yeah.
How you chosen that ratio of curcumin and
The gentamicin to the curcumin, you mean?
yes, ma'am.
Yeah, that was according to one of the papers that we had referred to. So there they showed that it had good effect. I mean, there the ratio was taken that way. That is 1 gram of curcumin with 0.1 gram of gentamicin. We also tried another one or two ratios, but we had to get back to this because this was the best ratio for the inhibitory thing that we had done. So.
You didn't try with QBD or something like QBD software or something?
No, we did not do that much because, basically we were not very sure because how we are going to be able to manage the curcumin, and also when you consider these herbal things, so even the last part of the work is still left. That is, we want to redo it with QBD. Also, we wanted to do the in vivo, that is the drug content in vivo, which is left. So we will be continuing it in the as the next work when we are going to be doing.
Okay. Thank you. Thanks, Matt.
Hello, madam.
Thank you.
Hello, madam.
Any other participants? Questions, please.
Hello, madam. Hello.
Hello.
Hello, ma'am. Hello, ma'am.
Hello. Someone is
Yes.
asking the question, but voice is not clear.
Yes, sir. Hello. Madam, am I audible?
Yes. I'm not able to hear you though.
Okay, ma'am. I will keep it in the chat box. Please.
Yeah, you
answer
can
the
put it
comment.
in the chat box.
Yes.
Chat box.
Good morning, madam. Am I
Yeah.
audible?
Yes.
Actually, madam, I had one question regarding
Yes.
the irritation test of the ointment.
Regarding
Did you
the
perform it some irritation test?
irritation test.
Yeah. Was that performed on animals or normal skin?
We took the liberty to do it on both. We also applied it on our own because anyways gentamicin or curcumin for that matter would not cause any harm. And of course the excipients that we use also are very common excipients. So we did it on ourselves, our own hands. And also we applied it on the animals that, I mean, we had not taken ethical clearance because we were not sure we were going to do this. So we just took it on the animals that were available for people who had got it for their own experiments, but it went well. So it did not have any irritation either on us or them. So
Thank you. Thank you.
yeah. Yeah, we had 3, and like I said, gentamicin was there, amikacin was there, but the availability of gentamicin was, free. Like we got the sample for gentamicin, and therefore we stuck to gentamicin. And also gentamicin is a broad spectrum antibiotic, so we decided we will go with gentamicin.
Anyone other has any question?
Yeah,
Anyone else?
I'm not able to hear you. Can you put it in the chat box? Okay, try again. Maybe I can hear you.
Okay, ma'am, this is Chaitali Lad here from the
Yeah.
ATAL Pharmacy College.
Yeah,
Just
Chaitali,
asking
tell
the
me.
participants if anyone is having any question or query. Okay, so I am just having the opportunity to extend the vote of thanks to you.
Okay, please. Yes, thank you.
So thank you very much, ma'am. It was a very nice presentation from your side. You had explained a very well session in a detailed manner. Since I am also from the pharmaceutical biotechnology department,
Okay.
we had already gone through this kind of the preparations, formulations, and you had explained very well the analytical and characterization part. Thank you very much, ma'am.
Thank you.
A very nice session from you. Thank you very much.
Thank you so much. Okay. So can I leave the meeting now?
Yes.
If nobody has any questions?